Yes, we can hear you. You can
Thank
start
you,
now.
sir. Thank you, sir. Thank you so much, sir. S- Uh, uh, from site now is almost midnight, 2:30 p.m. a.m. So yes, this is Ashutosh Nath. Um, I'm from Boston, Massachusetts, USA, and I'm doing my PhD in organic chemistry and synthetic chemistry at the University of Massachusetts, Boston, in Professor Zhang's lab. And today I'll talk about the in silico investigation on the anti-cancer activity of selected 2-aryl derivatives against the breast cancer. So this paper, first author the Moliani, she is also PhD student in University of Massachusetts Amherst, and Ajay Kumar, he did senior lecturer uh, at European University of Bangladesh. My today's presentation overview is the background about the breast cancer, then in silico study, then drug design, and after that, I'll talk about my work, my planning work, and then my work result and discussion, then final conclusion. So basically, the breast cancer, according to the World Health Organization, 2021, breast cancer became a most second major cause of the death in Western countries. And it also now 23% of all cancers death in Asia, also in the second leading cause of the death in the Western countries. Asian countries, and more than the 60% breast cancer in the Asian countries is cause of the estrogen receptor alpha positive is cancer. Basically, ER means the estrogen receptor alpha positive cancers. So ERs played a crucial role for the breast cancer development in the normal mammary gland and development. And also the sex hormones can contribute this the breast cancer. And on the other hand, the breast must be concentrated to the increased number of fat cells, cholesterol, and etc. So basically, the above is the 55 or older, the women are affected in the breast cancer, and it's almost this one is more common for women, not for men. And in 2022, accordingly, almost 2 lakh 850 new cases in invasively breast cancer will be diagnosed. So the family history also cause for the breast cancer. If the mother or previous the grandmother has a breast cancer, so it's possibly like to next generation to possibly the breast cancer. So and highly the breast densities for women for the fatty tissue. So yeah, and the heavy drinking, alcohol use for the cause for the breast cancer. So for the breast cancer treatment, the scientists, our researchers, the measure two obstacles. First one is the metastasis, and second one is the drug resistance. So metastasis is a obstacle because of the some drug not properly work, and skull is the when some cancer cell growth in body in any position start. So this called the metastasis. And for drug resistance is called the drug is not work properly in our body. That's why drug resistance is very obstacle for the breast cancer treatment. So that's why these two causes which are linked with the fatty acid metabolism. So when compared the normal cell and tumor cell have a wide range of significant difference there, the metabolic activity. So fatty acid synthesis is the key, the enzyme, the androgens fatty acid synthesis. So that's why blocking the FASN could be good way to treat breast cancer that has spread to our of our body. That's why as a drug development process, as a pipeline, first we need to the drug identification and drug validation, then lead identification, then lead optimization, and finally preclinical test. So after the preclinical test, we can go the drug approval. This is the another same things, just I showing the two slides. First we need to target identification, then lead optimization, then preclinical trials, then clinical trials, then finally the drug approval. So as far as rule, I started for drug identification for the in silico study. That's why I selected some synthesized molecule, the oxazoline. Oxazoline derivatives. This article published in the Journal of Organic Chemistry, Organic Chemistry in 2020. So this article showed some small molecule synthesized, and we collect this structure from the article. Also, we synthesize in our lab, in my lab, for the next preclinical trials. But firstly, I collect these molecules, almost 35 molecules, collect these papers. This is the my selected molecules, and then I collect from some FDA approval drug for against the breast cancer. So then I will compare with the FDA approval drug and my selected synthesized molecules. So and then I collect some proteins, because our targeted is the protein. So just a couple of slides ago, I told that the fatty acid synthesis is the main target for our reduce the breast cancer. That's why fatty acid synthesize I need to just select the fatty acid synthesize the protein. That's why 3TJM is the protein data bank. From the protein data bank, I collect 3TJM protein. This function is the acetylate metabolic process, fatty acid biosynthetic process, fatty acid metabolic process, fatty acyl biosynthetic process, and etc. Also, I select another three protein 3ERT4OR2J6M, the similarly for the cause of the breast cancer. My work that here, after screening almost 35 molecules, I found three best molecule in my research. Hit 4, I named this one hit 4, hit 6, and hit 8. First we now result discussion. First we did the molecular docking. I use for this the docking for the Schrodinger software. Is the best software for the molecular docking analysis. Every pharmaceutical company, basically in US based, Pfizer, Moderna, everyone that use the Schrodinger software for drug screening. So this software I use for the analysis for the docking. So left side is the my targeted 9 best hit compound, and right side is the FDA approval drug compound. So I see here that the docking score is better showing than other than FDA approval. We can see the hit 2 and hit 4, hit 6, and this FDA approval only FDA 1 is better other than, and my our compound shows that the 2, 4, 6 is better, and 8 also is better than other. And also I analyze the binding energy with the protein and my molecules. So this table showing the I select first the FDA 1 drug and analysis. I found the -42, but hit 4, hit 6 also better than the FDA 1 and other than the hit 8. So why we did the prime MMG GBSA analysis, because this is the best proof the binding energy. This calculation is fully the computational work. This I use for the prime version 4.8 Schrodinger Mestero 31 point version UMass Boston license software. And then I analysis the interaction with the my molecules with the protein, and found the best interaction with the amino acid. Those are my targeted, and those are the FDA approval drug targeted pocket. So because my time limit, I don't want to talk more this slide. And then I did the molecular dynamics. So why did we do the molecular dynamics? Actually, the molecular dynamics is predict the my molecule and the protein when the interact and bind, how long this is stable. So we can see this peak. This is the fluctuation at the time. I did there the 15 nanosecond. So we can see the heat 8 is too much fluctuate, while the starting point is the 1, but fluctuates too much, and high is the 3 point, almost 3.4. So this is too much fluctuate is the heat 4, but heat 8, but heat 4 and heat 6, we see that is not too much fluctuate. That means my protein and my molecule is bind strongly. It's not a loose binding. So it's binding energy is good. And also right side is the residue. This 3 protein, 3 dark residue is the same thing. It's not difference, but left side the RMSD value, root means a standard deviation value is showing the heat 4 and heat 6 the better than the other heat 8. After that study, we study the toxicity risk assessment. This is the body toxicity, because we need to drug like properties of this molecule. So that's why we studied the toxicity risk assessment. So we found that 4, 6, 8 also the better result than other than also 3. Here is the likeness properties -11, but other result is not good. That's why we select the 4, 6 and the better result for the toxicity risk assessment. And then the ADME properties. ADME properties is the absorption, distribution, and metabolism and excretion the drug. So the first one is the log P, the molecular weight. IC50 rule means we need to add drug below the 500 molecular weight, and also tropical TPSCA, hydrogen bond receptor, hydrogen bond donator, non-respiratory bond, molar defectivity. So everything showed that the heat 4. Also here is showing the good result in the heat 9, heat 5, but all result is not showing the good result. That's why because of some here the TPCA is the 117 is too much, because below 140 is okay, but it's too much. So that's why we consider the heat 4 and heat 6 and heat 8. Then the lipophilicity study PCI 50 value. This is the prediction value. This is not the experimental value. So this IC50P, IC50 value is the lower for heat 5, and heat 8 also. Heat 7 is the higher, and heat 4 is the too much. So IC50 is the lower than is good for a drug. So finally, my conclusion, my work focus on the anti-cancer activity, 2-aryl Oxazoline derivatives, and breast cancer inhibitory fatty acid synthesis, and my compound is the high potent, and it's comparison with the approval drugs, and using repeat scroll everything, we found that heat 4, 6, and 8 is the much better the bioavailability, and safety characteristics are low toxicity and non-carcinogenic properties, my compounds, and we assess when the molecular docking and dynamics, also the MMGBA, energy, binding energy, and found that the docking value is the 10.3, 10.1 is the best binding is the -7 up, that is -8, -9, -10. So -10 is better result for the drug in silico study. So this requirements that my research conclusion that next step we can follow the in vitro test and free clinical trials and clinical trials. So we already doing this work in my lab, in my professor lab, UMass Boston. We are doing again synthesis the compound, and we are trying to do using the preclinical and clinical test. So this work is still working. This work is not stopped. And this work just last month published this paper in the Journal of Biomolecular Structure and Dynamics. After 2-month review, this paper accepted finally 3rd October 2022. This is reference, and I'm very thankful to Molly Rani. She's a PhD student, UMass Amherst, USA, and Ajay Kumar, senior lecturer, European University, Bangladesh. And also I thank to Professor Jang Research Lab. This is my research group. Thank you so much for giving a chance to talk. Any question?
So thank you. Is there any question from the audience? So thank you very much for your patience
Thank you.
presentations.
Thank you so much.